So I'm going to pick up, this is probably a couple slides back, just to kind of get us back in. B cells are some of our adaptive immunity. Just keep in mind the difference between innate and adaptive is innate is not specific to certain pathogens, not much at least. Whereas adaptive immunity is very specific and it requires you to have encountered, well, it doesn't require you to have encountered something before, but it can respond more quickly to something that your body has encountered before. And so you adapt your immune system as you encounter new pathogens. Your innate immunity, your first line and second line of defense, they don't adapt. Like you don't change your body temperature, right? You don't change the way your skin functions or your sweat pH and acidity, salinity. See what I'm saying there? But we will with adaptive immunity. Just realized something. I don't think. I have no idea if it's picking up. We're going to hope so. For those who aren't here, oh well. Because if they can't hear this, then they won't know I'm saying it. And if they do, it works. So we're good. Okay. Now with B cells, one of our two lymphocytes that we talked about, I want you to look again at this thing. So here is the mouse. There we go. So here we've got this thing jutting out of our cell membranes. Okay, out of the B cell itself. Okay, so it literally is a cell, just like it says. This you'll see this red thing, and it's got two kind of arms that go outwards. Those arms are variable. That means they have different shapes attached to the end. These are made of protein. And to some degree, they are randomly produced. Now, the point of this is to make enough different shapes in the hopes that when they come in contact, when these cells come into contact with a foreign invader, that shape, whatever that shape may be, one of them on some of the cells will bind to a piece of a foreign invader. That's called the epitope. Does that make sense? Because there's proteins that stick off of invaders as well. Okay. Or some other shapes. But the point is if they are the right shape, then these things over here that our cells made, our B cells, if they fit perfectly, they'll bind. Okay. And that's how kind of how antibodies work. Kind of what we're getting at. So we've talked about antibodies. Those are not attached to a cell, but these are similar, except they're attached to the B cell. Okay, same kind of idea with this binding region and an epitope. Okay, so this makes B cells very specific. If it doesn't have the right shape, it won't fit, it won't attach, it won't recognize the invader as an invader. And each B lymphocyte has multiple copies of that B cell receptor. So every cell has one kind of receptor. So another cell could have a different kind of receptor. And of course, that means you're going to have multiple cells, right? Multiple B cells. And there are two variable regions. Like I said, those two arms that kind of point outwards like that Y shape. And each of the BCRs, those B cell receptors, are going to recognize only one epitope. Now, cool thing is, there are millions of epitopes they can make. So what does that mean? There's millions of foreign shaped proteins that they can attach to. So they're really specific. And antibodies, like I just said, are very similar to those BCRs on the outside. So what am I trying to get at here? I'm not trying to get you on this so to remember, BCR, the word for that, but just that B cells have a piece on them. Partially, we have enough that we're together for class. Yeah, at least I think I am. I'll find out if it's working. I found the stuff, and
yeah, everything should be in that bucket.
I just don't know if I have it all together correctly.
Okay.
I've got it together where I think it works, but I don't have that one attached. That would be easy, but
Sorry to interrupt.
no, you're fine. I have video and I think I've got sound, so we're good.
Okay. Sorry.
No problem.
I just know one of the classrooms.
Okay, not surprised. Thank you. If you're wondering, MSMS had an open house this weekend. That's why there's a piece of paper back there. This is MSMS stuff on it. And they used all of our classrooms in the building. So they removed all of the technology from all of the classrooms. And y'all are that 8 o'clock class, so. Okay. So y'all see what we're getting at, right? So this is an antibody. Doesn't that look just like what we saw on the outside of the B cell? This one's just not attached to a cell. Okay, pretty easy, right? So antibodies and these receptors on the outside of B cells function the same way. They have these variable regions and they are able to bind to specific things, all depending on if the right one comes in the right contact. Do y'all see how that's a randomness though? That's not controlled by the body. That's one thing I think that we kind of forget. We're like, ooh, I have the right kind of receptor. I'm going to send that cell right over there, but that's not how that works. How do you think it works? Hey, Shelby. Yeah. So if the body's not sending a specific antibody or a specific B cell, how do the right ones come into contact with a foreign invader? It's random chance. As long as you produce enough of them, they're going to be flowing through the bloodstream. They'll come into contact as they come into contact. And then once the contact is made, the body can start responding because it knows that something is there. Okay. It's not like we have cameras or a trip line that you can set off a warning device. For us, we have almost like bodyguards wandering around the body. Bodyguards that can only notice one certain kind of thing. But if you have enough of them, then those bodyguards will eventually come into contact and catch you. Catch the foreign invader. Does that make sense? I don't know the best way to really describe it better than that. But I think that randomness is something I don't think that we really talk about. We're like, oh, specificity, but our body can't tell it where to go if it doesn't know where it's at. It has to run into it by chance. And so the only way that you can get B cells or any of these antibodies and things to run into them by chance is if you have enough of them flowing through the system for them to come into contact. So people who are immunosuppressed, their immune system's not producing as much of these things. So how can they respond if they don't have enough bodyguards watching out? Does that make sense a little bit better?
But what would you like for people for someone to have a high B cell count?
Probably there's a chance that, okay, I'm sure, not sure if it's 100%, because this is not my forte, but I think a lot of what's going to happen there is that means that someone may have an infection. So like their body is, so once you start recognizing which one, those are going to start reproducing. And so you'll increase a B cell count of specific kinds for that reason. So when you find a white blood cell count, like we were talking about with leukocytes, or here we've got lymphocytes, those are probably an indication that someone has an infection that you may not know about. Now, does that discount the possibility that their body just normally produces a high amount? That's a possibility. Because just because we have a normal range doesn't mean everybody falls into normal. But does that make sense? Those would be the two major things that I would think of. Primarily, it's probably an infection that may not presenting any symptoms beyond high blood lymphocyte count. Yeah. Like for example, my niece right now, one of the twins, in her case, it's white blood cells. Her white blood cell count is, as of last time I heard, about 700. Normal is about 450. She was up to 900 at one point, and we don't know why. So we're watching this. She's going to go to a hematologist in early next week sometime. So we're trying to figure out, is there some kind of infection? Or there's another thing that bodies can detect, and it's not necessarily a foreign invader, but cancer would be one that can sometimes pick up something like white blood cell count. So remember, white blood cells are leukocytes. I'm not asking you to remember the leukocyte thing, but those are different than the lymphocyte, the B cells and T cells we're going to talk about here, but they do some of the same stuff, the way they detect things. So we're really hoping nothing's up with my niece, but for some reason her white blood cell count is higher, and there are no symptoms presented that we can tell. So if you find someone who's got a high count, there's a chance that some kind of infection or something that's not going right. With a third option of that may just be normal, but that's a much less possible than an infection or something like that. A long answer. I talk too much to answer y'all's questions. I could have done that faster.
So how is like if you donate, it's not plasma, but it's no, not bone marrow. It's some kind of, if you donate something for someone has cancer and they increase your white blood cell count before you donate, and it's like twice or like it's extremely high, more than what you used to.
Yeah.
How does that not affect you? Like I know that you're gonna donate it all out, but
Yeah.
like how does it not affect you before you do that?
I mean,
I forgot what it's called. But it's like you increase your white blood cell count like
Yeah.
first shot like week before.
yeah, I understand. I'm trying to think. I don't know exactly if that, because normally I think of bone marrow, but doesn't sound like what you're talking about. It might be a type of plasma, possibly.
Yeah.
May not be just regular plasma, but maybe a type of plasma, because that's usually the components. But either way, whatever it is, your question is to do with how does it not bother you?
Yeah.
And I'm sure in some ways it does, but it's a short increase and then decrease. It's not a, remember the difference between the acute and chronic situation that we talked about? Acute won't hurt you because it's your body's response for a short period of time. The chronic is the issue. I think that because more than likely your numbers are going to come down, obviously, because they're going to extract some from your blood.
Yeah.
And even if your count is higher, your count should start to go down because your body should stop making it because you won't have that stimulus. Now, if you were to stay doing that,
Yeah.
I do wonder if you might have issues with clotting, inducing blood clots from those kinds of things. That's I'm not sure about 100%, and that may not be exactly connected, but I know that some of those immune system problems, that's an issue. But probably for a week, and they take it, and then your numbers should come back down. That would be similar to a regular infection, I would think. Does that make sense? You have a question?
Okay. So is this the same like the tumors? So like the J cell count, if you have a tumor, even if it's benign?
Only if your body is responding to it. And that may not always be the case because it's yours. But your body can respond to cells that are producing like they're not supposed to. Now, a benign tumor, you may not respond that way because those are not metastasizing, they're not spreading throughout the body. It'd be one of those like metastatic cancers probably that are get picked up. I don't know fully, but I don't think benign tumors. I mean, I have benign fatty tumors. So I've got little ones throughout my body, and it's just adipose tissue that grows in nodules, and not that big a deal. I don't respond to them. The biggest problem with this is I think it's putting pressure on a nerve, and I need to get it removed so it stops, because I sometimes feel a weird thing going up this way, especially a fat finger right in that spot. And I was with the doctor the other day, and he was like, that might have something to do with some of these other problems you've been noticing. And I don't get it all the time, but I'm not like, this is not spreading to other places. It's just something that my body does.
How is that?
Go ahead.
Like, how does that even happen?
I don't know, actually. I really don't
Like,
know.
you just have adipose tissue that just builds up. That's what I think, like, I guess if you have it in a certain spot that just builds up, but it doesn't build up anywhere else and doesn't spread anywhere. So like, what causes that specific?
That I don't know. And a lot of times, I'm not sure that we actually have, I haven't gone into the research to see, but a lot of times we don't know that information yet. That's still stuff that people are studying. I actually have more than one. That's just my biggest one right now.
Yeah.
But I don't think that it means that it's spreading.
Yeah.
It's just something that's probably genetic,
That's what I was about to ask.
and that I don't know if there's an environmental factor that kicks in certain patches of adipose tissue, bam,
Yeah.
or if it's random, it just happens to turn on. But I have smaller ones. I have a little one in my lower back that sometimes I can feel. I've had one removed. That's how I know what it is.
Yeah.
And you can just see a tiny scar. I mean, it's almost like unnoticeable that I even had it.
Yeah.
So, you want to know, it's kind of cool. I watched them take it out.
What about with people?
Surprisingly, it didn't bother me as much to watch as I thought it would be.
So is it like just like a little yellow
Yeah,
fatty ball
yeah. Now they sent it off
to make sure, like,
to make sure that there wasn't any kind of
Yeah.
cancer going on. So I'm pretty certain that the rest of these are going to be the same way.
Yeah.
But it was kind of cool to watch it out because the yellow fattiness that we saw,
Yeah.
more closer to the color of the fat in our. Did y'all see fat in salamanders? Some of the other ones had the other class had some. It's a little bit darker yellow, not as dark as what I would have seen there, but it was darker than what we saw on the sheep heart. Just kind of a bit more buttery colored.
Yeah.
It's probably a little too much information, but it's kind of fun. Okay. So we kind of already talked about what this does. When we talked about some of that neutralization and opsonization that we talked about before. So here's the idea. Remember how I showed you that diagram of the 5 ways that the little antibody little things can attach? And sometimes they can surround it. Sometimes they can actually kill it directly. Sometimes they just attach to one and then attach to another one and another one attaches to that, and it causes a big clump. Same thing's going to happen with your B cells. Okay. Just like you would with those antibodies. So you have neutralization. So it binds and it kind of prevents it from being able to do anything to you. Opsonization, where these B cells bind, and then it causes phagocytosis to occur. We get oxidation, where it kills directly. Agglutination, where one binds to this one, the other side of the cell maybe binds to another one, and then another one that binds to another one. And then it causes them to clump up so much that they kind of just, the word is precipitate. When something precipitates out of solution, it becomes so solid that it can't stay floating around and moving. Okay, now this could become a clot maybe in the right situation, or it just basically stops moving and the body starts to remove it because it's not moving around. And then it can actually do, you can get natural killer lymphocytes. Okay, those are the same 5 things that we showed in that diagram in the last PowerPoint. So same situation, just with a cell attached. Those are the Y-shaped things. That makes sense? Okay. So here's that picture. Actually, maybe it was this picture here that we showed it, but you can see how they attach. So it's kind of wonderful. Our immune system is really cool. Okay. So y'all feel okay about those? Otherwise, I want to move on. Okay. There's 5 different classes of antibodies. I want to point out some of these only, okay? So again, remember this class, I'm not trying to be that specific, but I like showing you how things work. So IG, so you see those letters IG, immunoglobulin. So we've seen that word a couple times on slides. And so it's immunoglobulin M, immunoglobulin G. But we just call it IgM, IgG, IgA, IgE, IgD. Okay, so these are versions of antibodies that can be attached to either by themselves or are attached to your B cells. So IgM is the first antibody produced. So if once you get infected, IgM is the first one that your body makes. IgG is going to come later. It's the most common, and it's going to be your longest lasting antibody. So it's going to work the longest. IgA is really cool. I like this one because it's associated with body secretions. Now, can y'all think of a really cool body secretion where having an immune antibody system excreting out with that secretion would be a good thing? Yes, I'm saying it that way just to see if y'all can catch what I'm talking about.
Sweat.
Okay, maybe. But where is it most beneficial that someone excretes an antibody? Are you trying, well, the question is, in those situations, are you trying to catch, are you, so are those places where you're trying, this is so funny. Are you trying to catch an infection in those places? Maybe. So why would you excrete an antibody?
Release an infection?
Why would you, let me ask you this way. Can you think of any secretions that you give to someone? Oh, that's true, but I'm gonna call that a secretion in this case. This is gonna get weird. I'm gonna have to tell y'all the answer. What about breast milk?
Oh, wow.
Y'all didn't even go there, did you?
Yeah.
So it's a secretion that is purposefully given to another human. And one of the ways, funny, one of the ways that a mother can support a brand new immune system that's not able to produce a lot of that immunity itself early on. So IgA is a type of immunity, a type of antibody that a mother passes on. That's why I'm not too worried about COVID with my niece and nephew, because they're all breastfeeding. They're getting their mother's milk, even if it's from a bottle at some point.
So why is breast milk better than formula?
It comes with mothers.
Because of that.
That's a big thing. Also, that breast milk is perfectly tailored to that child, their needs.
So like, it didn't have an allergic reaction.
Unlikely. It is possible,
Yeah,
actually.
I was gonna say you could still find
There
that.
are some allergic reactions. There are some children that have allergic reactions to their mother's milk. Awful situation. And they have to go to things like some kind of formula that's produced. Yeah, it is possible. I would always suggest, for this reason alone, that breast milk be what you give to your child, whether you actually do the breastfeeding or if you pump. And so a lot of times my sister pumps and saves the milk in the freezer or the refrigerator if it's really short term. Won't get into more details of that because she probably wouldn't like me to.
But I've always heard that breast milk is better than
Yeah.
formula, but I've never known, like, I knew it was better for you and the baby, but I just never knew.
So, okay, for you, oh, now if you're asking for you, now it's energetically very, very expensive to do breastfeeding. It actually, it is taxing, mentally, physically, emotionally, everything you have goes into that child, from what I've been watching. But it also reduces body fat. So for the mother, breastfeeding helps to pull off some of that baby fat that came with the pregnancy as well. And if, since you are female and may go through this at some point if you decide to, I would suggest talking with someone who's like certified, helping breastfeeding mothers with a lot of stuff. So we actually have one of the very few people, if not the only one, certified this. I forget the exact certification, but she's in Columbus. She's very active on Facebook. I know her through the farmers market, and I got her hooked up with my sister because my sister was concerned about being able to produce enough milk for 2 babies.
Yeah.
It wasn't so bad when you had one. You can make a stockpile. In her case, they were basically drinking it as fast as she was making it. And when she went back to work, because we don't have a long period of time off in this country, and one day, I'm hoping we can fix that. We need 5, 6 or more months. I think mothers probably should take a year or so, but we're not at that point. Other countries are ahead of us there. But that would reduce that problem of being able to try to pump. So she has to pump at work, take it, put it in the refrigerator. The other day she forgot breast milk bags. And so thank goodness she works in a hospital. So she went down to the NICU and she's like, hey guys, you and my twins, I need some bags. And they're like, yeah, I want to see pictures. It was really cute. That would be the name, yeah. But this one is certified, like, I'm not wrong. She's certified in something that very few people in Mississippi are certified in. Yeah, she's really interesting. She's been here, I don't know how long she's been in Mississippi, but she's not from Mississippi originally. I know that when she moved here, I think there was only one in the state. Crazy enough, that person worked in this building. I found out this recently. And I think when the other lady came in, the first one maybe stopped doing that kind of consultation. But she has a lot of like tips and stuff on how to take care of breast milk. Side note, it's kind of interesting. She posts a lot of things when hurricanes occur because she talks about how to protect your breast milk if the electricity goes off. Because it's precious. And it's not something you think about. And it's a precious commodity. In fact, there are places you can donate breast milk as well. Yeah, I'm going to tell you something really funny since we're talking about this. And I know a woman here in Columbus. She was a surrogate mother kind of recently. And she's a big proponent of vaccination with COVID. And she's very outspoken, especially on Facebook. And she posted something one day which made me just laugh. I thought it was so funny. She said she's going to do some guerrilla warfare against people who are anti-vaccination and start handing out coffee creamer. She just had baby, remember? And guess what's in that breast milk because she's vaccinated? IgA. She called it guerrilla warfare. And it has to be one of the best things I had read all year. Not ethical, okay? Don't do that. Don't give people breast milk without telling them. Yeah, that chocolate pie, forgot about that. We talked about last time. Yeah. IgE here is one that responds to parasitic infections and allergies, just like we can also see eosinophils or your lymphocytes. Sorry, leukocytes, the white blood cells. So you can kind of tell where in an infection you are based on which antibodies in highest production or what kind of antibody is being produced. Does that make sense? So I think that's really kind of cool. Now there's IgD, but at this point, I've not read anything. We don't know exactly what it does, but it's got the right configuration to be an immunoglobulin. No. What do I want you to know? There are certain ones that come first. There are certain ones that are for secretions, like breast milk, for example. And there are certain ones that last longest, and there are certain ones that are for parasites or allergies. Now, do I need you to know which is one, which is which? I don't think that that's fair because those are really easy to get confused. Those letters don't match up to anything to help you remember. So I'm more interested in how they work than which one specifically do what, because you can always look that up. Does that make sense? Okay. Okay. So here's what they kind of look like. I'm gonna bend down for a second. My leg is hurting. So here is an IgM. You can see it's definitely very different. It's one of the first ones produced. Now, why do you think it might be like this as opposed to that? Why, if this is the first one, why would it have a different setup than this one?
The one that like would be the one that makes up the rest. Or like since it's first, it has to be bigger, and then the other one's kind of break.
Well, they don't break off because they're structurally different a little bit. Okay, that's what makes them IgM versus IgG, or IgA or so on. It's surrounded, right? Imagine if this was moving through your blood system. How would you imagine it moving around? Like a tumbleweed, okay? Wouldn't it more likely, if it's got something on all sides, come into contact with your infection first? Does that make a little more sense now? So I kind of got this picture of it rolling. Whereas if one of the others is moving around, is it possible they could miss if they don't get the right orientation? But IgM, on the other hand, is set up kind of in all directions, or at least in a lot of directions. Okay. Now T cells. This one's really interesting. These are produced in your red bone marrow and your thymus. They circulate in the lymph and the blood. And we're going to try to talk about what is different about them. Let's see how many slides I've got left. Okay. Okay. T cells are important, especially with COVID right now, because these are ones that are going to help us fight infections in ways that we don't yet understand. From things I've been reading, yes, we're producing antibodies, like here with our B cells and so on, but it's the T cells that are actually having a stronger effect sometimes that we don't fully understand. So for some people who don't get COVID the second time as easily, or they get over it faster, it seems that T cells have some part to play in that, but we don't yet understand what. So we've got this really strong portion of the immune system that we don't fully understand completely on how it's doing it. We know how it works. We don't know exactly what its function is with COVID. Okay. Let's see. Guys, I'm sorry. Give me a second. I have to go to the bathroom again. It's Friday.
It is very
I said so much has to happen.
I know.
I'm sure.
Don't have to ask the question. Then like an hour.
What, 2 minutes? CFP?
I forgot who the biggest is. Like 16?
Who's your,
Vanessa.
you have like, your last for me?
I mean, I answer questions, but.
But like, when you answer questions, is it like one sentence or two?
It depends. So like, if it gives me a lot of information, I try to answer thoroughly.
Yeah.
If not, I'm just like, because it has a place where you don't see something you could not observe, and then you can like write small.
But I heard that if you have, you know, piece of, you have to write it down.
I mean, yeah, just do your best and be thorough.
Oh, yeah, I wasn't just gonna like,
Yeah,
put one word in.
I know you.
But yeah,
Yeah, just be thorough.
yeah.
Is he your advisor?
No.
Oh,
So is it who's your advisor?
but yeah.
And mine's Ms. Cameron.
Okay, cool. She may be over here after the mission.
I think it's your age, so. Did you take it at, where did it, at state? Okay. Is it like you decide on the GRE website?
And it gives you like, whatever you have to take, I think.
So I kept looking and I got, like, you can take it at state, I'm like, no, I can't find it, but I just found it. And that's why I was wondering.
Make sure you take some, it be some old.
Oh, yeah. Do you take on your computer or your,
There's,
theirs? So you don't have to bring anything up like that?
yeah, it'll be too.
It's over.
Sorry, guys.
You're fine.
Okay, so a difference between B cells and T cells, you know how B cells had that Y-shaped thing and they directly attach. T cells don't do that. They don't recognize those epitopes, those different shapes on the foreign invader directly. They bind to an extra protein thing. And their purpose is primarily not to bind the invader, but to bind to a cell that has an invader inside of them. So they actually act against your own cells that have some abnormality, maybe an abnormal surface protein. Okay, so do you know of anything that happens to get inside of your cells? What kind of infection? Bacteria have their own cells, right? Because bacteria are cells. You know of anything that's not a cell that gets into your cells? Yeah, exactly. Jimmy Neutron. Awesome. That's exactly it. So what you've got real quick is a viral infection. So here's your cell. And let's say you've got this, virus. It attaches. And eventually what it will do is it's going to inject something inside of that cell. Okay, so what does it inject? It injects a nucleic acid. Now, what are our two options for nucleic acid? Don't have nucleic acids? So A, C, T, and G are the nucleotides that make up a bigger thing. So what do those make up? So you're right. So RNA is one. Or, well, M would be a version of this. Well, any RNA in this case. What's the other one? The DNA. Exactly. Okay. Now, more than likely, this RNA is going to be something like an mRNA. You know how we pass on information and we do protein translation from that RNA. What that's going to do is this virus is going to put in its nucleic acid. And depending on the virus, it can be this or it can be this one. But the cell will read these two things and either go through transcription and then translation, or just go straight through straight through translation and make more viruses. That's how viruses work, by the way. They use your cells to make more of themselves. They hijack. Sometimes I feel like they're holding a little gun to your cell. Just stick up. Give me all of your machinery to make more of me. But what they do is they kind of trick your cell into making more. And then you end up with more. And guess what happens? Boom. A lot of times your cell ends up what's called lysing, releasing all of these out to go infect other cells and do more of that reproduction. But they cannot reproduce themselves, so they hijack your cells in your body and use your cellular machinery to do this process. Does that make sense? And that's the kind of cell that the T lymphocytes, the T cells are going to be protecting. And y'all see how this is connected to COVID now, since SARS-CoV-2 virus hijacks your cells in this kind of manner. And there seems to be some really major power behind what T cells can do to protect us. The problem and the reason why we don't understand it very well is, from what I understand, T cells are hard to isolate, so you can study them. We know what they do, we understand how they, we know they exist, but it's really hard to put them in a controlled experimental setup and make them do what we think they're going to, experimental group, control group. It's hard to get them to do that. So look, that looks a little different binding site, doesn't it, than the Y-shaped one. And I'm going to pick up here next time. I think this would be a good spot to kind of pick up, because clonal deletion is kind of a fun thing. I probably won't test y'all on this, but it is kind of cool to see how it works, because our bodies are just really amazing. We'll finish up with our material on Wednesday. I'll see you on lab today at 3:30. I have a Zoom meeting at 2. I don't think it'll take an hour and a half, but if for some reason I'm trying to get off, just know I am coming. I'm just down in my office. Yeah, I think that's it. Wednesday, we'll finish up this. We've got like 10 more slides left, and I'm not adding anything new slide-wise. The only thing we'll do is we'll talk a little more about COVID and how, just try to use it as an example for immunity and how vaccines work, to kind of put that together. And that'll be the last of your material. Okay.
And
We have a test Friday, unfortunately.
like,
Well, that's what I want to use the vaccine thing for.
it's not like, you don't have to tell us exactly what kind of it's from, but like our slides, there's more information than what we're supposed to
Yeah.
remember. So like,
Let's do that at lab today.
can we do that last?
Let's do that lab. Let's take one.
And see. what
Yeah,
we
let's
really-
go through everything. We'll just start at the beginning, because it's really only 3 sets of slides.
Yeah,
It's quite a bit of info, but
but I just wanted, I didn't want to spend so much time on one day and I didn't need to necessarily. spend
let's do that
much
at
time.
lab. And then that way we can finish this and vaccines on Wednesday. So we'll do that at 3:30. Okay. If no one's using this room, we'll start over there, but if not, we'll come in here so we have all of this space
Okay.
to work with. And we can project everything up. Okay.
That works.
See you all 3:30.